For day 7's homework, there were 2 questions. The first one was trying to take a chemical signal, and that signal causing expression of protein X. By expression, we mean producing the protein from gene X. So expression of gene X would get you protein X. And then protein X acting as a transcription factor to cause 2 genes, gene Q and R, to be produced, and one of these, protein Q, would be a secreted protein. So let's start at the beginning. And for this homework, we have our chemical signal binding to a receptor. It's a G protein coupled receptor, actually 3 subunits, but I normally just draw 2 just to make it faster. Binding to an enzyme, so when the conformation changes here, it binds to an enzyme, and that produces a second messenger. I would have preferred to see this as cyclic AMP, as this enzyme is commonly adenylate cyclase, but there are other possible ways to do this. And then, as I showed in the video, this shows cyclic AMP, the second messenger, binding to a transcription factor. Here it is. And allowing RNA polymerase to bind, we get messenger RNA from gene X. So this is gene X, coding region, and regulatory region. So the messenger RNA you get from gene X is messenger RNA for X, which goes through a ribosome. Protein comes out of that ribosome, and that is protein X. Protein X is another transcription factor. It then moves into the nucleus, binds to an enhancer binding site on gene Q. So we get messenger RNA, and then through a ribosome, we get protein Q. And same thing for gene R. In each case, it's noted these are enhancers. Remember, I also wanted to repress gene Y. And so here's our cyclic AMP binding to a transcription factor down here and repressing gene Y. Here's another example. External signal, G protein coupled receptor. Here's adenylate cyclase, producing faster just to write cyclic AMP, made from ATP. Binding to a transcription factor, and here's the combination. Now, the way it would actually work, because second messengers don't last very long, so this would actually be a kinase that would take an inactive TF and an ATP and make the active TF with a phosphate covalently bound, and that's what would bind. That lasts longer. The phosphate stays bound until a phosphatase takes it off. Here's gene X, messenger RNA from gene X, protein X, the heart shape. Don't need this. The protein X is itself a TF, so it binds to gene R, binds to gene Q, messenger RNA from each, protein R, protein Q, and then those go on to do whatever. Notice also our transcription factor is a repressor, so it's blocking gene Y. And this person drew it nicely. Here is RNA polymerase, and they actually put it in the way, which conceptually is basically what it's doing. It's preventing RNA polymerase from binding, although often it would be somewhere over here. Here's another example. Here's our external signal, G protein coupled receptor. Good to have more labels on here. Here's the second messenger being produced, binding to a transcription factor or activating phosphorylation of a transcription factor. Here it is, binding to gene X. We know it's gene X because here's the messenger RNA coming out, going through a ribosome. Here's gene X. Here's protein X from gene X. And it would be repressing for gene Y, which I don't think is shown here. Then gene X into the nucleus, binding for the yellow and the blue. Yellow is P, blue is Q here. I wanted to show this one because there's a common source of confusion with eukaryotic versus prokaryotic cells. In prokaryotes, you can have a single transcription factor binding with multiple coding regions. So this one transcription factor in a bacterium might cause the production of multiple proteins. That's how the Lac operon works. But in eukaryotes, it would be a separate stretch of DNA for each. Here's gene Q. We would need our transcription factor, RNA polymerase, messenger RNA, and that would be for Q. And separately, gene P. Again, the TF binds. Here's RNA polymerase moving along, and we'd get to protein P. Or sorry, yeah. I think I may have said R originally. Another example, just to show you there's lots of ways to do this. Here's our receptor, second messenger. Here is gene X in purple. And then protein X. It's the TF, here it is. Didn't need to show a second protein here. This green transcription factor X would have been enough. To gene Q, gene Y, and then our original second messenger combination is repressing gene Y. This one showed everything really nicely and completely, and I think was likely useful for the person who did it, but I just want to point out that you could make it more efficient on an exam. So they've separately shown the input, receptor binding, the shape change of the receptor here. It's gone from this shape to this shape. The G protein separating, affecting an enzyme. Here's our second messenger. There's the G protein affecting the enzyme. And then that combination causing RNA polymerase to bind, making messenger RNA for X. Here it's shown repressing gene Y. Here's the repressor for gene Y. And here's TFX now, once it's produced. That is activating RNA polymerase to make messenger RNA for Q and for P. There's an error here. This would be gene Q because that's the protein it's making. That's one I didn't worry about here, but I did want to correct it. Here's another example. Sorry, here's the next question. We want this particular cell, in this case, to be expressing G, which is in turn going to cause expression of M and N. So how do we get gene structure of G so that it's only going to be produced here? So this person identified V and segment posterior. So they said the gene structure has to include ventral and segment posterior. But in fact, it would take one more, which I hope most of you are getting now. It would take HOX2. So this area has HOX2. If we only do V in segment posterior, that would also get this cell, might also get this cell. So all of these have V in segment posterior. But only this one also has HOX2. So we need to add HOX2. And then all three enhancers, required. That then has RNA polymerase moving along. We get protein G. Protein G, again, does not need to show a second part here, just there's protein G, and good to say enhancer, and RNA polymerase moving along. This example showed something else I liked. What I liked was it showed all genes in all cells. So every cell has these genes. This person labeled them 1, 2, 3, 4, 5, 6, 7. And over here did a key. Segment anterior, posterior, ventral, dorsal, HOX1, HOX2, and HOX3. So in this particular cell, only the ones in blue are expressed. And that would be 2, which is segment posterior. So it should be segment posterior. And 3, which is ventral, and 6, which is HOX2. Therefore, if there's a whole bunch of genes here, A, B, C, D, E, and so on, then some of them will get only one of the transcription factors that they need. And so they won't be expressed. So this cell, for example, this gene C needs 2, 4, and 6, but it only gets 2 and 4, the ones in blue that I highlighted in yellow. So it won't be expressed. However, this gene down here, it gets 2, 3, and 6. So it's getting segment posterior, ventral, and HOX2. And so it gets expressed. That makes it, this person decided, gene G. So here's the G protein, and then G protein binding to an enhancer region. Best to say this is enhancer. Here, best to say, actually important to say, requires all 3. Otherwise, I don't know that you know that. And so we're going to get RNA polymerase going along for M. Again, M and N should be separate. So which cell is this? It's going to be this one. This is the region of HOX2. That is segment posterior. Also here is segment posterior. And then this area is ventral. So this cell has HOX2, segment posterior, and ventral. And it's the only one that's going to express this. Another example. So here is G, gene G, ventral, segment posterior, again, HOX2 would be required here. And that got you protein G, and then here's G binding here for M and N, getting you the messenger RNA and then the proteins.